Okay, good morning. So today we are doing a lab on primary cell isolation. Sound good? So I'm going to talk to you about some of this stuff, about isolating cells, about why that's important, how that fits into things that we've already discussed. I have a handout here with the procedure on it. There was also a procedure that was posted on Moodle last couple of days. It's a revised protocol from Thanda on what you guys are doing on doing the growth curves, cell growth curves. How's that been going, by the way? Cell growth curves. Is that interesting? Yeah. Good. Okay. So I think she wanted to include a spin step in there. The other thing that I'd like to give you is a sheet that has some suggestions about how to keep a lab notebook. Just a couple of quick things, like you might want to have write a purpose, methods, results, conclusion. Not familiar? So even though I'm handing you a protocol for this with a procedure on it, in your lab notebook you should create an entry that describes an experiment. And I have kind of an interesting mix of priorities with the lab notebooks. I think sometimes in some classes they emphasize them too much, and then people end up making them very pretty and neat and copying things over so it looks better and all that. Whereas I kind of go the other way on that. I don't want it to look necessarily pretty. I definitely don't want you to copy anything over. I mean, whatever you write at that moment while you're doing the experiment is your lab notebook. Copying things over, that's no longer a lab notebook. And the reason for that is because your recollections are the clearest at the time that you do the experiment. And writing something over, you actually can dilute the content of what you had written. In order to look neat, you leave out the calculation, whatever, that you needed in order to come up with the right amount of reagent or that kind of thing. And so then it becomes more difficult to reproduce the experiment because you've copied it over. The calculation is no longer there. You get rid of a lot of important information. So the most information is really there right at that moment. And it's most akin to a journal. Some people will keep a little book by their bedside or whatever, and write in there every day what happens. I mean, it's an interesting exercise. You go back and you look at, well, what was I thinking? What was I feeling? Earlier on at some point in time. Well, that's the way a scientific lab notebook is. It's like what happened at the time. It's a record of that moment, and to copy it over some number of days later is to dilute, possibly damage the record. So you don't want to do that. Okay. And Rachel, you wanted to take a makeup exam. When do you want to do that?
Can I do that after?
After the lab. Yeah, okay. That way, I guess, though, we're not going to discuss this quiz. But we can discuss it some other time. It's pretty straightforward. Although I wasn't satisfied yet in how you were doing on the quiz. And the reason for that is that science is very specific, right? Do you notice that? There's if I ask you what body temperature is, it's 37 degrees centigrade. So if you say it's 36 degrees or 35 or 40 or something, that's close, but that's not good enough. If you were a medical doctor and you take someone's temperature and it's off by a couple of degrees, it's like, they're still living, it's kind of within their range. And there's actually a lot you can learn from having the correct temperature. And likewise, the reagents that we're using in combinations with growing cells. 37 turns out to be an important temperature. If the temperature was a few degrees higher than that, the cells will go into heat shock. So they're used to being maintained very specifically at that particular number. So therefore, on a quiz, if you're a degree or two off, then that's a point or two off. And so that can make a difference between B plus and a B or whatever. And so you want to make sure that you get these details. And the interest in the details, it's interesting because in many human efforts, details are based on who's in power, who's popular, who's been voted in or whatever. In science, it's just a fact of nature. You really can't go against Mother Nature. That's the way the universe is set up. Sorry, it's made out of atoms. Sorry, life is organic material. That's the way it is. You might want to have some other belief. That's fine, but you're not going to be able to pass a biology class with some other belief. So, okay. All right. So today we're going to talk about isolating cells, and it's an area that I think is actually really interesting too. One thing to view the perspective of all this is that so all life is made from cells. And it's a pretty powerful statement. So therefore, cells are pretty interesting. So what is a cell? A cell is just something that's enclosed. Some living protoplasm enclosed in a membrane, right? And if it's a bacteria, it's also enclosed. I mean, every living thing is enclosed. And that's because there's the outside world, and the outside world is not living. Right? There's living things around, but the outside world is non-living and potentially threatening. And then there's the inside. Inside of the cell, it's protected. And so, life sort of exists inside. It's an enclosure. It's a controlled environment, controlled chemistry. So that's why the idea of a cell is so fundamental, and why isolating cells is kind of an interesting thing. If you want to study life on any level, animals, humans, plants, anything, you have to know something about cells, and we're going to be isolating cells today. So I think cell isolation is kind of the first step, right? So at least experimentally, isolate cells. First thing you do. Second thing you do, you can grow cells. Sort of a sideline of growing cells is you can freeze cells. Freezing is good for long-term storage. So if you look at these important things, we've been growing cells, doing our growth curves, getting an appreciation for how cells grow. We've learned how to freeze cells. Now we're going to isolate cells. It's actually like the first step, isolation of cells. Okay, some more perspective on this. What kinds of cells are we going to isolate? Well, we had touched on stem cells already, right? We've talked about that a little bit. Kind of interesting stuff. Trying to get you interested in, understanding why this class is kind of mainstream. Why stem cells have become very mainstream in biology, important in medicine, and there's a lot of money behind it, right? There's the $3 billion California bond measure. It's just our state alone. It's willing to commit that kind of resources to trying to understand what stem cells are doing. Okay, so where do stem cells exist in the whole thing? You've got the fertilized egg. Fertilized egg, and that's kind of the beginning of everything, right? Well, the embryonic stem cell is kind of the equivalent of the fertilized egg. Wouldn't you say? Right? Because the stem cell gives rise to all the other cell types. In fact, if you isolate an embryonic stem cell, could you make an embryo? What do you think? I was saying it's the equivalent of fertilized egg. So therefore, if we isolate one of these embryonic stem cells, is that going to give us a new individual? Yeah, it is. It's going to give us a new individual. So, I mean, that's been done in, experimental animals, mice and things. So we tend to not do that with humans because then you get into the cloning thing. many George Bushes or Michael Jacksons should we have? I don't know. But embryonic stem cells are functionally equivalent to the fertilized egg. So then, what's the next step? Well, obviously there's a bunch of cell divisions and such, but I mean philosophically in terms of lineages of cells. So you've got a fertilized egg. That's a stem cell. There's another type of stem cell. So when I draw an arrow, I don't mean to say that one cell, goes to the other. It's sort of a philosophical discussion, right? This is not cell division I'm drawing here, okay? But you've got the adult stem cell, or sometimes people call that a lineage stem cell. So what's that? This is a cell that reason why it gets the stem cell name is because a bunch of cells can be derived from this cell, right? It's a lineage stem cell, so it's not as primordial as the embryonic stem cell. And if you implant this in the uterus, it's not going to give rise to a new individual, whereas this will, right? So, but these cells are really crucial in the body, all over the place, right? Because they're pools of these primitive cells that will give rise to a couple of different subtypes and help replenish that organ constantly. In one area in the body where, multiple areas, but the areas where there's a lot of replenishment required is the epithelia. Because the epithelia gets worn off, right? Your epidermis, for example, in the skin, you're rubbing against stuff all day long. Your skin is sloughing off, they say, right? So there's, house dust is mostly human skin, all that powdery stuff that appears everywhere. That's your skin shedding. And so, those cells need to be replenished. And same thing with the lining of your gut. You're eating food, right? And it's going down your whole digestive tract. The whole digestive tract is coated with epithelia, which is similar and related to the epithelia on the outside of your body. And that epithelia is seeing a lot of food material that is going to cause it to slough off. It's going to cause some damage or whatever. As it goes through the digestive tract, some of the epithelial cells are going to go along with it. So then it needs to be replaced and replenished. So whenever you've got this requirement of making lots of things, you need some stem cells. And the blood is another obvious one, right? We're constantly refiltering our blood. Cells are taken out. We lose blood at a continuous level, at a continuous rate as well. So we need to constantly replace blood cells. And there are stem cells that are in charge of budding off new blood cells constantly, as a constant requirement. So stem cells are really critical. But adult stem cells are different from embryonic stem cells, the fertilized egg. And why is that? Well, these are multipotent, but these stem cells are pluripotent. So pluripotent means, like they'll do everything. But multipotent means they can differentiate into a bunch of different cell types, but not into everything. So there are actually stem cells in the skin, right? And I was saying that the epidermis is constantly being sloughed off, so then you're going to have to replenish that. So there's got to be a pool of stem cells somewhere. And there is. And, we're going to be isolating some cells today, and we'll be, in the zone where there are some stem cells, and we're going to try to tease them out. Okay, so what happens after the adult stem cell thing? And by the way, are you guys adults? How about a 5-year-old? Is that an adult? Yes. Biologically, there's only, fetal, and then there's adult. So once you're born, you're an adult, biologically. There's requirements on the system, right? Once you're a free floating organism, you're an adult. At least that's the terminology used for a lot of the cell work. Okay. So what happens now? Well, there's a bunch of cells that could arise from this
original cell. We'll just draw 3 just to make the point with this bunch. Okay, there's a bunch. And these cells, at this point, this is this idea of commitment. In other words, these cells are going to become some specific kind of cell, but they don't look like it yet. But they're committed. So this concept was overemphasized when I was taking biology a couple of decades ago. So it was the idea that cells terminally differentiate, and that idea was so emphasized there was no possibility of turning back. Once there was a skin cell of a certain type of fibroblast, then it could only be a fibroblast, and keratinocyte, the epithelial cells, could only be that cell, couldn't do anything else. And everything was very rigid. That was convenient for people dividing their departments into who's got what expertise and all that stuff, but it wasn't reality, actually. The level of commitment, there's a bit of plasticity in the system. There's a bit of flux. So cells can become other cells, and I think especially within a certain niche in the body, if there's damage, then the body is able to draw upon cells that happen to be around there, and kind of recruit them in and get them to do other things. It's all part of your body. I mean, why make a rigid category, right? Why not say, hey, we've got a pool of this kind of cell. Is that good enough? Yeah, let's use some of those cells and try to reset up the vasculature, try to repair this wound, at various levels. So I'm interested in that because I see a bunch of that in my own research too, this plasticity idea. Okay, but then there's beyond this point, you get the classic differentiation. Okay. I guess I'm going off of screen here on the movie. So I'll draw it down here. Okay, so after commitment, you're going to get differentiation. And that's where a certain cell type, starts to look like that kind of cell. Like a cell that's kind of elongated and spread out. The fibroblasts look like that, right? There are other cells that are sort of jagged looking. They might have long protrusions or something like that. A neuron. There's more cobblestone type shaped cells, kind of rounder but not exactly round. The keratinocytes. So there's differentiation that occurs, and then that explains where we get all the organs and systems of the body. So today's cell isolation, where are we in this grand scheme? Well, we're starting out with a biopsy, a chunk of human skin. We also use dog skin for demonstration purposes. We can isolate cells from various parts of the body. We actually were interested in chondrocytes at one point. So we went down to Petaluma and got some calf meat. Sort of a recently slaughtered calf, and they gave us this chunk of meat. And then we, dug out some of the cartilage and digested up the cartilage and freed up these chondrocytes. And then we could grow them in culture. It's kind of cool. So, you can grab anything and isolate cells. I think a lot of times in research, there's an emphasis on using one particular cell, like a particular branded cell. Do you notice that? I don't know. HeLa cells. You ever heard of HeLa cells? There's a woman, Henrietta Lacks, or Lane, who had cervical cancer. And they isolated the cells from cervical cancer, and that cell line has been going on forever. So that gets back to what we were saying before about cell lines versus primary cells. So our cell isolation from parts in the body, are those cell lines or primary cells? They're primary cells, right? Okay, so so in the absence of a source of cells, if you can't get an official cell line, if you can't order them if you can't afford to order them from a company, or there's legal limitations on what cells you can get cells? Absolutely. Every living thing is made out of cells. Nobody's going to stop me from working on cells, right? I mean, you just need to get a sample and digest it up and tease out some cells, and then you're good. Okay, so in the overall scheme here, where we are today is we're certainly not at the fertilized egg thing. We have a biopsy. So this fertilized egg is differentiated. All the cells have differentiated. They've all gone to at least the adult stem cell level. So we're not going to isolate any of these, right? But we could be isolating some of these, the adult stem cells, because we have a skin biopsy. So there, potentially should be some adult stem cells. There could be some cells that are not quite committed yet, but will be giving rise to further differentiation, and then there will be differentiated cells. So the fibroblasts that we're, accustomed to working with, those fall in this category here, differentiated cells. Also the keratinocytes, differentiated cells. They're not just committed. The committed cells in the skin, there are cells that they call transit amplifying cells. And those give rise to a lot of the further differentiation of the epithelium. One of the things about skin, this is a kind of a specific point, but the epithelium of the skin is a complex epithelium, because they sometimes call that a stratified epithelium. So what does that mean? It means that there's the epithelial cell, but then it has further layers, right? It keeps dividing and giving rise to more and more epithelia. So, the layer of our skin, we've got, about 10 layers of epithelial cells. It varies based on body location a bit, but, it's sort of on average, say it's about 10. So the epithelial cells become more and more differentiated as they move to the top. And at the very outside, those are actually dead squames. A lot of those cells are already dried up. They form a kind of a tough little bag thing, and they're on the outside of the skin. So then it doesn't hurt if you rub yourself, which is a good thing, right? We're just sloughing off dead cells. That's why, people exfoliate and stuff. They want to expose the living cells to look more beautiful. But, very quickly, after you exfoliate, those cells are going to terminally differentiate, and then there are going to be a bunch of dried dead old cells on there anyway. But, sort of the normal thing. Okay, so we're going to be isolating adult stem cells, committed cells, and a whole bunch of differentiated cells, because they're mostly differentiated cells if we just get it from the skin biopsy. Okay, some more philosophical thinking here on cell divisions. I think this is kind of interesting, because you guys are doing growth curves, right? So is there some limit to cell growth? Can you keep going? Week after week, you guys have come in here several weeks, and you've been counting cells. And you're going to do that again today, because we do that as a kind of an ongoing part of the lab. So we're doing the new topic, but we're also constantly doing these growth curves. And it's because growth curves are so essential. Cells growing, what's more fundamental than that? It's even more fundamental than looking at them in the microscope. Looking at them in the microscope, you might see something morphologically about them, but the fact that they're growing and dividing and taking up room, that's kind of an essential quality. So growing cells are a big deal. But is there some limit to how many weeks we could go? You've got your dish there. You trypsinize them. You take them out of the wells they're in. You move them over, dilute them out, move them over to the next set of wells. Could what week are we in at this point for the cell growth curve? 3? Yeah. Okay. So we're out 3, right? Could we go out 6? Could we go out 10, 20, 30? At some point, how long are these cells going to grow? Well, there was actually debate about this years and years ago, and there was one guy who was saying, well, cells in culture will live forever, and then, that kind of got to be the accepted idea. It's like, well, cell culturing was kind of new. It was thought, well, it's just the miracle. If you can grow cells outside of the body, they're not affected by all the crazy things we do to our bodies or whatever. We've got a controlled environment. Those are immortal cells. They can keep growing. And this guy had a culture growing of some cardiomyocytes, I think, and they were, the culture kept going. And he had it going for like 34 years or something, didn't see any end in sight. So it seemed like, they were immortal. But then another guy came along, Leonard Hayflick, who's a local guy, and I've actually met him. A lot of these guys, a lot of the scientists are kind of interesting personalities. They're not really rock stars exactly. Looks like a guy you'd meet in a bowling alley, actually. But Leonard is a funny guy because he's got a good sense of humor, which is good. And he came up with the idea that cells stop dividing after a certain number of cell divisions. So primary cells taken from the body, they only go a certain number of replications. Well, that would kind of explain why we get old and die eventually. It's like, you're only good, your cells are good for a certain number of replications. And then that also kind of makes sense when you start thinking about the DNA. Well, the DNA is this complex genetic information, it's this code. How many times can you make a copy of that complex code without making errors? In the environment, we've got high energy radiation coming in and all this stuff. There's a lot of what they call insults to our DNA. There's a lot of damage that's accumulating, right? So it would make sense that you could only go out a certain number of divisions. So anyway, so Hayflick came up with this idea of the Hayflick limit. And basically, you isolate cells initially, and they need to get going in the culture, so the number might dip a little bit, but then they're going to take off in cell number, and then they're going to plateau. They're going to plateau out. And if you've got like 10 to the 6 cells here, you go up to like 10 to the 18th cells, and this is a log scale on the cell number, right? Just like we're doing in the growth curve. So this is not a daily growth curve. This is sort of the lifespan of the cell growth curve. And you look at the number of weeks in culture. Well, there's going to be, this plateau at this point. This is going to be a finite cell limit. That means that the cells are not going to grow infinitely. They're finite. There's a certain number of cell divisions, a certain number of weeks in culture, where at that point the cells are going to stop dividing. So yeah, we're fine week 3, probably fine, a couple more weeks, 10 weeks we could go out. But, start to go more than that, you're going to start to approach this finite cell limit. Depending on how dilute you make the cells, how many population doublings there are. So it's not chronological aging, but it's a replicative aging. Kind of an interesting concept, right? So chronological aging is, well, this guy standing up here is good for maybe 80 years or whatever like that, right? But on the other hand, there's the cell division thing. How many replications can the cells go through? And if you force them to keep dividing, which we do in culture, right? We've got these culture dishes, putting trypsin on them, and we're splitting them out onto a new culture dish. Well, you're forcing them to divide. It's like divide or die. So we're giving them lots of nutrients. They've got lots of amino acids and sugars and all the stuff that they need to grow. And so they'll keep going. But they'll go out to this finite cell limit. So there is this one interesting thing. There's this finite cell limit, but that's for primary cells. That's for normal cells in our body. So what kind of cell would do that? Would take off like that? So it starts to plateau, and then suddenly you see in the culture, a subset of cells start growing rapidly, and they don't stop at all. What does that sound like? Cancer, right? And that happens. That's actually an observation. You can culture primary cells, and at some rate, you'll get spontaneous formation of cancers, just like in the body. As we age, there's more replications required, And it actually is not, as I said, it's not necessarily chronological. There are patients that have some kind of defect, you know, a mutant protein or something. It was a disease we were studying in the derm department at Stanford, epidermolysis bullosa. And you know, there's a couple of different forms of this, but you know, on all of them there was a protein that was defective. So there's one amino acid that's either the wrong amino acid was put in there, or the protein is truncated, or something happens to the normal function of protein. So then the cell is affected by that, and the cells are being sloughed off, let's say, in the epithelia way too quickly. Well, then those adult stem cells need to keep dividing. They're going to divide, you know. And a certain number of cell divisions, they're going to start to peter out. So, and they'll do that, but then, you know, these cancers start arising. So, I mean, you can have a patient that's, you know, a 5-year-old, and they can start developing cancers because there's a defect somewhere, and so their tissue is being damaged so quickly. The cells have to keep replicating to keep up with that, and they've gone to a certain critical limit in terms of possible cell divisions, and cancers start to arise. Kind of interesting stuff. So, I mean, this biology that we do here, the fundamental stuff you see with cells, that, you know, extrapolated equals what happens in the clinic. You know, you see patients, and, you know, there's genetic disease, and there's various sorts of diseases, but, I mean, a lot of this is explained on just even a cell level. So you get a lot of insight into medical ideas just thinking about what cells do. Okay, so we were saying that a cancer cell would do this. So another name for a cancer cell is it's a cell line. Cancer cells are cell lines. So you go into almost all labs, like 90-something% of the labs that do any kind of biology on animal or human, and they're working with a cell line. And it's got a name. You know, it's the HeLa cell line, or 293 cells, or WI38 cells, or, you know, a whole bunch of different cell lines. And so the advantage to that is that the thinking is that if multiple researchers use the same cell line, and you get a result, and you publish that result, other people could reproduce your result. How? They'll get the cell line from you. Or we could even centralize the cell lines. Right? We'll store them in Washington, D.C., or Bethesda, Maryland, which is right outside Washington, D.C., which is where the National Institutes of Health are. That's the main governing body for health in the United States. And it's institutes of health because they cover everything. All sorts of disease at every level. There's a National Cancer Institute. There's a National Heart, Lung, and Blood Institute. There's all sorts of sub-institutes within the National Institutes of Health. And so, so you could get cells from the NIH, there's this ATCC collection of cells, so people bank cells. So you can get a particular cell that somebody used, reproduce the result experimentally, and you've got something there. That's the way science has developed in the United States for decades, right? And personally, because I was exposed to the medical field, we had patients coming in, they had disease, we were interested in understanding and potentially curing the disease. We started isolating primary cells. So that's why I've kept up with that practice of isolating primary cells constantly. And now that I'm so vested in this over many years, I also realize that the benefit of it is that we do experiments on a bunch of different types of cells. In other words, isolated from different patients, right? We're going to be isolating some skin cells, but they're from, Joe and Barbara, and actually not Barbara, so they're all males, apparently. Because we're actually using foreskins, right? So all these male skin samples that we have. But there's variation, patient to patient variation. So this is not a WI38 cell or a HeLa cell. It's not something you can go to the ATCC and get as a particular cell line. Those are cancer cells. We're not dealing with a cell line. The cells that we isolate have a finite lifespan, so they're going to peter out after a certain number of weeks. But there's a real advantage to this whole struggle on constantly isolating primary cells, and that is that if we see some phenomenon, and it's reproducible amongst a bunch of different patient samples, then we've learned something about like probably all the patient samples. You know what I mean? Whereas if you focus on one particular cell line, yeah, it's good standardization and anybody can reproduce your experiment, but you've learned something about that one cell line. Then you take what you've learned and you apply it to the clinic. You get some patients in and you say, well, we learned that this signaling pathway works this way, and we're going to try to cure your disease using that information. Well, you never learned anything about patient samples, though, did you? No, we had this primary cell. We had not a primary cell. We had a cell line, standard cell line that we use. Okay, well, that's a transformed cell line. So, you know, you're not used to looking at patient variation, are you? No. Are you used to looking at primary cells? No, not really. Well, then, what do you really know? So you go into the clinic and you do clinical trials, and you know, a bunch of the patients don't survive the clinical trial, or you know, or there's no benefit. Hopefully, you know, they survive. But you know, if they, you know, if there is mortality associated with it, you got to stop the clinical trial immediately, right? And actually, if there's no benefit too, a certain number of trials, you know, a certain number of patients involved, and you see there's no benefit, they'll stop the trial. So recently, they stopped a trial from a company called Geron. And it's interesting, you know, Geron's a local company in Menlo Park. You know where that is? Yeah, just on the So Geron, I really like Geron. They've done some excellent science, and I actually met Leonard Hayflick at Geron. You know, he was sort of hanging around, talking to a guy I know well, Mike West, who had founded Geron. So Geron had a bad result in a clinical trial. They had this thing called telomerase, a telomerase inhibitor, and they were trying to use it as a cancer drug, and it showed no efficacy whatsoever. So you got these patients with cancer, they're dying at a normal rate during the clinical trial. It's like, well, we should stop this trial because we're not helping them at all. So they did stop that trial, and the stock, you know, went down 60% one day. So then I was sorry that I got the stock, but I'm always interested in trying to get this research stuff to go. So what they had then was this telomerase. I mentioned this. So a telomerase inhibitor. How does that fit into all of this? You ever heard about telomerase? Anybody know what that is? No? Well, the reason for even bringing this up is because, you know, we need to understand something about the primary cells we're working with, right? So, our primary cells, we've accepted the notion that they'll only divide a certain number of times. So they could say that population doubling. How many population doublings? Not the chronological age of the cells, not that they're 80-year-old cells, but that they've divided a bunch of times. And how far will they go? Well, you know, if human fibroblasts will go like 60 to 80 cell divisions, and then that's it. So, why does that happen? PD, population doubling. Some limit to the capacity. Well, every time you get a cell division, there's loss of some DNA at the end of the chromosome. So, you know, I don't want to go into endless depth on this because, again, this is a lab, and I like to lecture. But I just need to give you enough background to truly appreciate what it is that we're working on. So what we're working on is these primary cells. They have a finite cell limit, a certain number of times, certain number of population doublings. I had mentioned that fibroblasts, they can go like 60 to 80 population doublings. And that's about it. And then why? And the why is because the telomeres short. Well, that's pretty profound. right? I thought it was because of the damage to the DNA. That's what it was, the radiation damage to the DNA. Isn't that what's causing aging? Yeah. Yeah, and there's a couple of contributors to the whole thing, and we're still trying to figure it out. There's a local institution really interested in this whole aging thing. What's that called? Buck Institute, right? They're sort of associated with the university. We have a graduate program with them. There's a lot of good opportunities, I think, at the Buck Institute. So they're interested in this kind of stuff. One of the scientists there is really good at doing these growth curves and stuff. Dr. Judy Campisi, and we try to collaborate with her. We have the same setup as she has, actually, the same cell counter. So when you use that culture counter Z1, that's what she's using too for her population doubling measurements. So the telomere shortened. So you're saying, okay, so all of those other insults from the environment, there's oxidative damage coming in. That's why people say, you should eat stuff like blueberries and stuff. They're antioxidants, right? But too much oxidation is going to be damage to your DNA and damage to the cell generally, to proteins and other things within a cell by oxidative damage. Then there's the radiation damage. Too much UV light. You don't want to get a sunburn. Why is that? Well, it's going to cause thymidine dimers on the DNA. There's DNA damage of various types occurring. Yet, nonetheless, the telomere shortening, that's the trump. This is sort of the ace in the deck. This is the thing that's really limiting the whole thing. So the cell has got, it's a cell and it's got a nucleus and there's chromosomes within the nucleus. And the chromosomes are condensed pieces of DNA, right? So DNA in the end is a linear polymer, right? It's these base pairs, and you draw it as the double helix. Well, there are two linear polymers that are running anti-parallel to each other, 5' to 3', right? That's what DNA is. It's a linear polymer. So then there's an end, right? There's one end and there's another end. It's linear. It's either circular, either the ends are connected or they're not connected. Well, in our DNA, they're not connected. They're sort of hanging out there. So the mechanism that's been used is that our chromosomes protect those ends by putting a bunch of repetitive DNA sequence at the end. Multiple copies of this stuff. Well, that's gonna, if there's any damage to an end, to DNA, if something comes along and starts nibbling, chews off some of the end, or if you get DNA replication, there's a constant nibbling at the end, the DNA. So then this protective sequence at the end of the chromosomes is going to be in trouble. It gets too short, and then that has implications for the lifespan. And basically, the cell can't survive if there's too much cell damage, too much DNA damage. There's actually a particular molecule that's the sort of gateway for all this stuff. P53. It's a protein of 53 kilodaltons in size, and it controls whether the cells are allowed to enter into the cell cycle and replicate their DNA. Right? P53 is the gatekeeper, and it sort of checks on the status of the DNA. Is the DNA healthy? Well, for the DNA to be healthy, you can't have these ends chewed up too far, and so P53 can detect that. And it's like, okay, so that they've been nibbled down too much, we can't replicate anymore. Shut it down. And that's where the population starts to plateau. It's like it's not going to go through any more cell divisions. P53, the gatekeeper, says, hey, there's too much DNA damage potentially here, so we can't replicate. Why would the body want to do that? Well, because if you keep replicating and there is damage, you're going to generate cancer cells. And then is the host going to live? Are you going to live if you've got cancer? No. So the body's like, well, we don't want to generate a bunch of cancer cells, right? We kind of want to keep the status quo going. We want to keep this person alive